Hello everyone, my name is Olga Pietrzyk. I work in the Neuroplasticity and Metabolism group where I'm pursuing my PhD. Today I would like to present an article titled Identification of AgRP cells in murine hindbrain that drive feeding. In this publication, researchers have discovered a new group of AgRP-expressing cells in the hindbrain and demonstrated their orexigenic function. I hope you will enjoy it. But before discussing the results, I reckon it will be helpful to give a brief overview of central melanocortin system. This neural network in the brain regulates energy balance, including appetite and energy expenditure. It consists of two main types of neuron in the hypothalamus in the arcuate region, and here we have AgRP neurons which stimulate appetite and decrease energy expenditure, and POMC neurons suppress appetite and increase energy expenditure. These neurons act through melanocortin-3 receptor and melanocortin-4 receptor, and balancing these factors is critical for regulating body weight and metabolic health. Interesting is that neurons expressing POMC and melanocortin-4 receptor can be found not only in the hypothalamus but also in the hindbrain in dorsal vagal complex. This complex consists the area posterma, nucleus of the solitary tract, and the dorsal motor nucleus of the vagus nerve. MC4 receptor is highly expressed in DMV, while POMC is expressed in the commissural NTS. Research has shown that these POMC neurons are related to suppression of appetite. Additionally, cells in the DVC can detect signals from the gastrointestinal system due to their expression of receptors for amylin, insulin, CCK, and GLP-1. I think it's also worth noting that AP is circumventricular organ and does not have an intact blood-brain barrier. So it seems that only AgRP neurons are missing element, and that's why researchers have decided to search for them. So, in order to confirm the expression of AgRP in the hindbrain, the researchers collect hypothalamus and hindbrain tissues from wild-type mouse, AgRP heterozygotes, and AgRP knockouts. Then they conduct RT-PCR, and the results we can see on the picture A, and confirm the presence of AgRP mRNA in both regions. PCR fragments corresponding to the size of the AgRP mRNA were amplified both from the hypothalamus and the hindbrain, but not in samples where reverse transcriptase were omitted, and no mRNA was found in the AgRP knockout mice. Additionally, real-time RT-PCR analysis showed that AgRP mRNA was expressed in the hindbrain of AgRP heterozygotes, but not in the knockout mice. Furthermore, and we can see it on picture S1, they observed that AgRP expression in the hindbrain was lower than in the hypothalamus. To locate AgRP expressing cell near the DVC on the mouse hindbrain, the researchers conducted single-cell RNA sequencing on cells isolated from the AP region of 10 days old mice. As we can see on picture C, they use the Evans blue for visualization of the AP region. On picture D and F here, we have cell groups identified in the samples based on the expression of signature genes that define each cluster. And the results on picture A here showed that AgRP mRNA expressing cells were grouped with GABAergic neurons, glutamatergic neurons, and tannicide-like ependymal cells. On picture G, we can see that subsets of AgRP cells express markers such as SNAP25, which is a pan-neuronal marker, SLC32A1, which is a GABAergic marker, also SLC17A6. This is a glutamatergic marker and markers characteristic for tannicide. And to compare these findings with adult mice, they analyzed the publicly accessible datasets from microdissected AP tissues, and AgRP expressing cells in the adult AP tissue mainly express SNAP25 with mixed population of SLC32A1 and SLC17A6 expressing cells. And all these results together indicate that AgRP expressing cells are present in the mouse hindbrain. We know that there is a significant and progressive increase in the expression of hypothalamic AgRP following food deprivation. Here, researchers provided that hindbrain AgRP mRNA expression was also stimulated by fasting in both male and female mice. Staining with AgRP antibodies was not detectable in ad libitum fed mouse, but became detectable after fasting. What is important, the signal was detectable only in wild-type mouse, and in case of AgRP knockouts, even after fasting, the signal wasn't detectable. These results suggest that hindbrain AgRP is responsive to energy deficit. Next, the researchers proceed by conducting a kind of examination of three independent mouse lines to analyze the expression of the reporters in mouse embryos and also in adult mouse. This involved an investigation into the localization of expression in specific regions and also within particular cell types. And as you can see on picture A, they start with AgRP-EGFP line. In this line, EGFP expression is possible only in cells with AgRP promoter. And they analyzed GenSat data and found that EGFP signal is detectable in mouse embryos. They also did some additional staining using wild-type and AgRP knockouts, and they found that AgRP signal is detectable only in wild-type mouse. Next, they check AgRP Rosa LSL TD tomato line. Here we have expression of TD tomato in cells with AgRP promoter, and they analyzed brain slices from 10 days old mouse, and as you can see, we have strong TD tomato signal from arcuate nucleus in the hypothalamus and also strong signal from AP region in the hindbrain. They also wanted to check if we will have similar effects in the adult mice, and as you can see in the same line, the TD tomato signal is also detectable in adult mouse. They also did some additional staining and confirmed that cells expressing TD tomato also co-expressed the neuronal marker. And the third line they checked, it was AgRP CRE-ERT2 Rosa LSL TD tomato line. In case of this line, we also have expression of TD tomato in cells with AgRP promoter, but we need to induce this expression with tamoxifen. And as you can see, also in case of this line, TD tomato signal was detectable after tamoxifen injections. And these results indicate that the expression of reporters in the hindbrain of three independent transgenic lines, it was AgRP EGFP, AgRP CRE, and AgRP CRE-ERT2, support the notion that the reporter expression is specific to AgRP expressing cells. Next, researchers investigate the impact of energy deficit on activity of AgRP neuron in the hindbrain using whole-cell patch clamp electrophysiological recordings. Following a 24-hour fast, neurons in AP and neuron in commissular NTS regions displayed depolarized resting membrane potential and increased action potential. Changes in resting membrane potential we can see here and here, and changes in the AP frequency we can see here. In contrast, neuron in the sub-P region showed no significant change in activity or membrane potential. These results indicate that AgRP neurons in the hindbrain can detect energy deficits, but also that there is a heterogeneity among hindbrain neurons in different regions. Treatment with monosodium glutamate leads to elimination of AgRP neurons in the hypothalamus. When this treatment is carried out on neonatal mice, it results in the elimination of 99% of AgRP neurons. And here, neonatal MSG treatment was performed on mice with expression of TD tomato in AgRP cells. And as you can see on picture A, it successfully eliminates almost all AgRP cells in the arcuate region, but not in the AP region. And that means we can study the hindbrain population without interference from hypothalamus. So in next step, and now I'm talking about the timeline on picture B, they decide to use AgRP HM3DQ mice. In this mouse line, only cells with AgRP promoter can be activated using CNO. So both control and mutant mice receive MSG treatment at postnatal day 2 to eliminate hypothalamic AgRP neurons. And after 8 weeks, these mice were injected with saline and their food intake was measured over 3 days. On the 4th day, they received CNO injections, and once again, their food intake was measured immediately. And as you can see on picture C, both groups has similar food intake after saline injections, but CNO injection significantly increased food intake and body weight in AgRP HM3DQ mice. So they decide to repeat the experiment in Klime's chamber to assess energy expenditure, and they found that CNO injections significantly increase food intake and the respiratory exchange ratio in AgRP HM3DQ mice, but there were no significant changes in the energy expenditure. As we can see on picture A and F, CNO induced FOST in arcuate region, but only in arcuate intact AgRP HM3DQ mice. After neonatal MSG treatment, in arcuate region there is no FOS signal. What is interesting, even after neonatal MSG treatment, we can still see the FOS signal in AP region. These results indicate that homogenetic activation of AgRP cells in the hindbrain stimulates feeding in mice, and it happened even when there is no AgRP cells in the hypothalamus. In the end, researchers wanted to see if activation of AgRP cells in the hindbrain would increase food intake. And for this study, they used an optogenetic approach with a light-sensitive protein called SOL, which was present in AgRP cells. And as you can see on picture B, when mice were not exposed on photosimulation, they consumed a similar amount of food as control mice. However, after photostimulation of AgRP cells in the hindbrain, AgRP soul mice showed increased food intake. I think it's worth mentioning that this optogenetic stimulation was possible only during the nighttime, and it doesn't work during the daytime. Next, and we can see it on picture C, AgRP soul mice received neonatal MSG treatment to eliminate AgRP cells in the arcuate region. At 10 weeks of age, arcuate lesioned AgRP soul and control mice underwent optogenetic stimulation, and it also resulted in increased food intake. These findings indicate that hindbrain AgRP cells promote feeding independently of AgRP arcuate neurons. Okay, so I have already discussed all the experiments, and in conclusion, I would like to leave you with the piece of information about the AgRP cells in the hindbrain. I must say that it was my first recording presentation, so probably I will look a little bit lost, but anyway, it was fun for me. Thank you so much for your attention, and now I'm waiting for your question on YouTube channel. See you on the Q&A session.